In this video of Introduction to Neuroscience, we just finished going through the passive properties of neurons, what happens when things are at rest. And I told you that the reason that the neuron maintains such readiness is that it's ready at any moment for something to happen. And so this is a really exciting lecture because it's the first one we're going to start thinking about what happens when something changes, when something is happening to this neuron. And after all, the things that might happen to the neuron are all the things that we care about, like what happens if you see something you didn't expect to see? What if you hear something that you didn't expect to hear? What if you feel touch that you weren't expecting to feel? How do you not fall over and respond appropriately to whatever it is that you're supposed to be responding to? So the active properties of neuron are starting from the same place that we started from last time, where we learned about the Ohm's law and how there is an RC circuit equivalent of this particular equation that describes how ions are maintained and flow from the inside and the outside of the cell. And this maintenance of the resting potential is the electrical basis of the resting potential and how the cells are ready when something does change. What we're going to do now is going to take the same equation, and we are going to look at what happens when we then artificially inject a little bit of current into the circuit. So we're going to be thinking about this. I'm going to explain it a couple of different ways. One way of thinking about it is that in terms of the circuit diagram, we're going to be artificially injecting a current. So what does that mean? It means I'm going to add a second clamp between the inside and the outside. I'm going to shock the heck out of it, like the equivalent of taking a key and sticking it into the wall socket, that kind of shock. That's a current, okay? That's the kind of current I'm talking about when I'm saying injecting current. It's not a syringe. It could be a syringe because syringes are metal. But anyway, it's kind of like if you took a key and stuck it into the wall socket. Don't do it, just an analogy here. So when you are injecting current into a cell, okay, the question is, if you injected a current, like in a square way, like the following way, you inject some current, you turn it on, then you turn it off, what actually happens, quantitatively speaking, to the membrane voltage of the cell? How does it respond to this injection of current? Now, as an analogy that's perhaps a little less contrived than my sticking the wall socket analogy, this current could also equally well be some kind of naturalistic sensory stimulus, like a flash of light that hits your photoreceptors, right? That effectively gets translated into an injection of current into your photoreceptor cells. So what happens to the voltage in that scenario? So we're going to derive that from Ohm's law, following up from last lecture where we derived how Ohm's law follows from the Nernst potential. So let's do it. We have Ohm's law here, but as you'll notice, there's a couple of extra terms because things are slightly more complicated, but just a little bit, so just bear with me. Here's what's going to happen here. We're going to write the membrane current, the total net current going through the membrane, I sub r, and it necessarily has to be the sum of the current going through each of these parallel pathways. Okay? So there's two pathways here. There's a resistive pathway and a capacitive pathway. And so we're going to count the current that's going through the resistive pathway as I sub r. That follows the same Ohm's law that we had before, where it's the conductance times V. All right, so far so good. We're also then going to count the current that's going to go through the capacitive pathway. And because we're injecting current across the intracellular and the extracellular terminals, and we're injecting current through a capacitor, the current is going to be determined by C sub m, the capacitance of the membrane, times dV dt, the rate of change of the membrane potential. Okay, the faster it changes, multiplied by capacitance, the more current is going to be passing through the capacitive pathway. This is the property of capacitors when we pass current across a capacitor. So this comes directly from theory that we have derived from electricity and magnetism, and it applies equally well here, except instead of a normal capacitor, our capacitor now is a lipid bilayer, but it acts as a capacitor in this case. So once we've written the equation that way, those of you who may like differential equations as much as I do may make the following observation, that this equation, despite all the subscripts and all the different stuff that's going on, it's actually a relatively standard differential equation of the following form, where we have dx dt equals ax plus b. So x here is the unknown, which is voltage, right? So it appears one time here in its native form, and it appears one more time here in its derivative. Everything else is a constant, because I know G and I know C, And I injected the current so I know what I external is, right? That's the injected current on the other side here. So I know everything except V, and I can express V as a function of dV dt. Okay, so I know how to solve equations of this particular kind. So I can just write down the equation from my knowledge of differential equations and how to solve equations of this kind. I can write down the solution of voltage and how it changes as a function of time in this particular form. Now, don't be intimidated by the form of the equation, because we can actually break it down piece by piece and figure out what's actually happening here. What's happening here is that everything outside the parentheses is actually a relative constant. We have I, my injected current, which I know, and G, which is the conductance of the cell. It's just the number of ion channels that are open. Okay, so this is good. This part's taken care of. And then we have 1 minus an exponential to the power of negative t divided by some other constants. I know how a function of that form can be drawn. It is exponent of that particular kind. It's going to be something that goes up exponentially and then stops after a little while. In particular, the rate at which it goes up, the curviness of this curve, how much the voltage potential responds to the injection of the current, is going to be proportional to what's called the time constant here, because it's the term that negative t is divided by. The time constant of the response is going to be the product of the resistance of the cell and the capacitance of the membrane. And that's a constant. We don't actually have to know what they are because we can measure it. And for most neurons, tau is going to be something like 1 to 100 milliseconds. And that's going to be how quickly the cell can possibly respond to an injection of current. The injection of current is nearly instantaneous. It's going to take the cell, depending on the particular cell we're talking about 1 to 10 to 100 milliseconds to respond to that change, how it's going to go up. The amount to which it goes up is going to be proportional to the magnitude of the current, because that's the first term up here, right? After the current is turned off, the cell is going to go back down to whatever it was before, and it's going to be an exponential curve of the opposite direction where it goes back down. In other words, we are going to see, in response to injection of current, some kind of exponential depolarization, because we're injecting positive current. So it's going to depolarize the cell, and it's going to follow this shape. After the injected current is taken away, there's going to be an exponential hyperpolarization in the opposite direction, where the cell goes back to something like the resting. We can derive all of these quantities and the exact magnitude of exactly what the membrane potential is going to do by computing a function of that form using constants that we can either measure or infer from what we know about the cellular properties of my neuron. Using this equation, we can then also reason through that the smaller time constants mean that V sub m can change faster. It means things can change faster. It means that I'm more responsive. It means that when something changes, I am ready to change my membrane potential really, really quickly rather than really slowly. Okay? And so this tau is then once again a function, right, of the density of the channels, how many there are per square area, the area that we're talking about, like literally how large is the cell that we're talking about in the local area that's responding to this change, and then also gamma, the single channel conductance. Now the single channel conductance is probably going to be a biophysical property of that particular ion channel type. I can pick different ones. They might have different conductances, but if I'm stuck with the same one, the gamma is not really going to change, but I can change the density of these channels. Right? And I could change that by choosing to express more protein or less protein. Okay, so these are some of the knobs that I, as a cell, might have the ability to change by changing protein expression. And I can also change protein expression by saying I'm going to express this type of ion channel that has a larger single channel conductance rather than this other one that has a smaller one, stuff like that. These are all knobs that cells can change in order to change and tune how quickly it's able to respond to an external stimulus, either a natural stimulus or an artificial stimulus, an injection of current of this type. And we can use these equations then to once again answer the same question we had before of what happens if there are not enough resting ion channels. What you can see here is that if there's not enough resting ion channels, that means the conductance is going to be different. The G is going to be different. pay attention to where G comes into that equation. Does it change how quickly the cell responds to external changes? Or does it change the magnitude of the response? Okay, answer that question for yourself by looking at an equation and try to figure out, just reason through it, okay? What are the physiological consequences of there not being enough resting ion channels? What does that mean? Which term changes? And then where does that term actually appear in my equation? And reason through what's going to happen if you're in a scenario where artificially, through some kind of pathology or some other type of manipulation, you don't have enough resting ion channels. So that's a question I'm going to leave you to answer. I'm going to move on and then talk about the fact that every single thing we have said so far in this part of the lecture and the previous one are under the approximation that my neuron is a sphere. Okay. In the limit of a spherical cell, everything I said has been true so far. But we know that one of the crazy cool things about neurons is that they're definitely not spheres. It's one of the really cool things about them. In fact, they're so much not spheres that they are the opposite of spheres. They are super long and thin, and their entire job, in many cases, is to get information quickly from one end of the body to a different end of the body. This is really important if you happen to have neurons that have shape, right? This is why they have shape. This is why they're not spherical blobs. This is a particularly lovely example of neurons not having spherical shape from the connectome. This is an anatomical reconstruction of many, if not most, of the neurons in the adult Drosophilid fruit fly central brain. And in this animation, you can see that through electron microscopy reconstruction of these volume, scientists have reconstructed all of these neurons, including all the different ways in which they reach far beyond their cell bodies and are very much not well approximated as spheres. This is a wonderful thing, but it also makes it really complicated because everything we've done so far has to do with things changing in time, but not in space, right? It's true for spheres, but what happens if you don't have spheres? And spheres are good for lots of things, but not good in the scenario where you're an eagle and you're hunting, right? Let's say that you are a neuron that's in the eye, and you spot a rabbit. Now, you have to change the exact pitch of your wing muscles and tail muscles, and maybe some of the ways in which you're pitching your feathers to dive down really quickly and get that rabbit before it runs away. It is really important that the information from the eye gets to the tail as quickly as possible so that you can score dinner. Spheres are not very good at doing that. In fact, spheres are not very good at doing that, and what we need are wires. In the same way that an airplane might have a wire from the cockpit where the pilot lives to the tail where you might have to pitch it in order to either go up or down when you're landing or taking off, same thing happens here. So neurons are the biological equivalent of wires that convey information, often very quickly and often over large distances, approximately as large as the animal is. And it does so by having a couple of different compartments where the cell body lives somewhere in the cell, in the animal, and there is a long process that's known as the axon. That is the output of the cell. That is when the cell is going to pass information from whatever it's gathered to wherever it's trying to go. And this long, thin region is called the axon. And so we're going to talk a little bit about the approximation of the neuron, not as a sphere, but as a cylinder, a long, thin cylinder. What happens if we consider that the cell is actually cylindrical in shape and might be really long rather than spherical? This is the point at which we've talked a little bit about changes in time, right? We talked about how voltage changes in time. So in calculus speak, that would be dV dt. This is the first time we're talking about changes in space. I'm just going to establish a little more terminology here to make sure we're comfortable. In terms of a cylinder, we're talking about the length of the cylinder. And so I'm going to, instead of using t for time, I'm going to use x for this axial direction of the cylinder, right? So anytime we're talking about change in space, how voltage changes in space, we're actually going to call that dV dx to differentiate it from dV dt. So similarly, there's a version of an equivalent circuit diagram that we can draw, just like the RC equivalent before, where here we're actually considering space, because now currents, instead of being able to just go between inside and outside, so here that's I sub m, the membrane current, currents can also flow along the axial direction of my cylinder, and that's I sub I for intracellular. So currents can flow inside the cell, and currents can flow through the membrane through channels. So it can go up and down, I sub M, and it can also go on the inside of the cell in the axial direction, I sub I. And we can kind of chop up the cell, the cylinder of the cell, into little segments, little DTs, okay? And draw this little diagram as it repeats on and on and on in the left side and the on side. So like, just not worry about the ends for now. Let's just only care about the middle of the cylindrical piece of neuron for now, okay? So if we do that, what we can do is write down, instead of Ohm's law, we can write down what's called the cable equation. And the cable equation is going to look a little bit more complicated than Ohm's law because it happens to be a partial differential equation. It's called a partial differential equation because it has a term in it that has dv dt, the change of voltage in time, and d square v dx squared, which is change in space, the second derivative of the second spatial derivative. And so the voltage is going to be a function of all of those things. Once again, for those of you who love differential equations, feel free to dig into this. There's lots written on the subject, lots of really cool models. Those of you who may not care about the details of this cable equation, we're just going to tease it apart a little bit. And once again, we're going to be looking at the parts of the equation where there are constants that we know, and there's things that change. So let's pay attention to just those parts for now. We're going to look at the cable equation, and we can see that it's separable in the following way. There is a set of terms that have to do with how things change in space, and a set of terms that have to do with how things change in time. And they're actually separate, which is great, right? They're not all mixed up together. It could have been worse. So the things that have to change in space is the second spatial derivative, and we're going to lump everything else that's a constant into this lambda square parameter. And this lambda square parameter is what's called the length constant of the propagation. It tells you about how much the voltage propagates in space, how much it spreads, right? If something is happening in the middle, your neighbors are going to hear about it, right? It's like if you're living on a block, it's how quickly rumor spreads from neighbor to neighbor to neighbor as everyone whispers to each other. If you whisper really fast, the length constant is going to be larger. If you don't whisper very fast, then it's going to be a little more localized. So the length constant of the propagation, lambda squared, is going to be the ratio between R sub m, the resistance of the membrane, divided by R sub I, the resistance of the intracellular space. The intracellular space is a salty solution, so it's definitely conductive, right? But it's not infinitely conductive, and so it's the relative ratio of how conductive the membrane is versus how conductive the intracellular salty water space is. Now here I'm using the notation that big R means bulk properties, and little r means small patchy properties. The patch is the property per square area, okay? So if we express it this way, you'll see a really interesting relationship start to emerge here, because the membrane resistance is going to be the resistance of each patch of it divided by 2 pi a, where a is the radius of this cable. Remember now, instead of having a sphere that is completely symmetrical, we have a cell that is cylindrical. So the cylinder can have different radii. It could be bigger, it could be smaller. Similarly, the axial resistance of the cylinder is also inversely proportional to A, but there's an A squared term in the denominator instead of just an A term in the denominator. If we do the division and cancel things out, what you'll see is that the length constant of propagation happens to be proportional to A. In other words, the larger the cylinder, the voltage spreads farther. We're going to plot that a different way. The other way, pictorial way of saying what I'm saying is that if there's a voltage in the middle, it's going to spread in space. Your neighbor's going to hear about it because those ions are going to flow to the left and right. The larger the diameter of the cylinder, the larger the diameter of the neuron, the larger the diameter of the axon, the more it's going to spread. This is important because having a larger axon is actually a really valid strategy if you wanted your message to go farther, faster, okay? But as in all things, there is a trade-off. It is not without cost. Maintaining a larger diameter of your axon, of your cell, also means that you actually have to pay for it metabolically. You have to grow it, you have to maintain it, you have to feed it, you have to give it ATP, you have to do all the things that you have to do to keep a larger cell alive. And the trade-off there is that you get an axon that is larger in diameter, which has a larger exponential decay length constant lambda, so that your message, your electrical message in the cell, is able to spread farther and farther apart. So these are the active properties of neurons. We've talked about how things change in time, as well as how they change in space, by reasoning through these physical analogies and looking at the equations that govern them, that we all know and love from physics. And so to summarize the basics that we talked about, are neurons responding to injected current by changing its membrane voltage. It changes so in an exponential way, with either exponential depolarization or hyperpolarization in time. We then talked about how the density of ion channels can determine how quickly neurons can respond to change. We talked then about the electrical property spreading in space. This is governed by the cable equation. And the cable equation makes a prediction about how the diameter of the axon in the cylindrical approximation determines the fastness and extent to which membrane potentials can spread. In the next section of the video, we're going to talk about we're going to layer on a little more activity, a little more excitability into the properties of the neuron, and finally get to the point where we have the thing that you're all here for, which is the action potential. That's next.